So here we are going to go over the Bradford assay. Now this is a technique used to quantify proteins. Specifically, it is a colorimetric assay, and it is essentially utilizing an interaction between a light-absorbing molecule and the protein of interest to understand what the concentration of our protein is. Because remember, when we talk about protein quantitation, we have two different categories. We have colorimetric quantitation methods, and then we have intrinsic quantitation methods. And the Bradford assay is a colorimetric protein assay method. Now, the Bradford assay is composed of this reagent, which we call the Bradford reagent. Now, the Bradford reagent has three subcomponents. It has this dye known as Kumasi Brilliant Blue, or CBB G250. And Bradford reagent is also made up of ethanol and phosphoric acid. Now, the purpose of this phosphoric acid is really important because as an acid, it provides that acidic medium needed for the Bradford assay to work. Essentially, our CBB G250 or our Kumasi Brilliant Blue dye, this is the structure of the dye. And we can make note that it has these two sulfonic groups and it has these three nitrogen groups. And in acidic conditions, when we have that phosphoric acid, in acidic conditions, our nitrogens are going to be protonated and they are going to have a positive charge, whereas our sulfonic groups are going to be negatively charged because they have a very low pKa. So in acidic conditions, when all our nitrogens are protonated and our sulfonic groups have a negative charge, the overall charge is a positive 1 because we have three nitrogens which are going to have positive charges because they're going to be protonated, that is +3, and then we have two negative charges, so overall we have a charge of +1. Now this positive form of the dye is more red in color, and so red is positive when we are in the positive form, so that's going to be in acidic conditions. Now the dye itself, so this dye, Kumasi Brilliant Blue, can exist in three different colors or three different states. The red, which we just talked about when we have the overall charge of +1. It can exist in a neutral state where the charges cancel each other out. And then we can also have a blue state, which is going to be negative when all the dye forms of Kumasi Brilliant Blue have an overall negative charge. So what's really important to understand is that these three forms of the dye exist in equilibrium in our Bradford reagent, even at low pH values. So these three exist in equilibrium. Now, of course, we are going to have more of this red form in acidic conditions, and we're going to have a small amount of the blue form, but they all exist in equilibrium. Now, what's important to understand is that the blue form has this negative charge, and what happens is that the blue form is what's going to go interact with our protein of interest. So the blue form of the dye interacts with amino acids, specifically it interacts with lysine and arginine, and then it also interacts with hydrophobic interactions with aromatic groups. Now, the interaction of the blue form of the dye with the amino acids stabilizes the blue form. Now, what's really important to understand is that if the blue form is going on to interact with the protein, we are going to have a reduced amount of free blue dye. So free floating blue dye, it's going to reduce because it is interacting with the protein. And remember, we have to maintain equilibrium. And according to Le Chatelier's principle, if something is reduced, we have to circumvent that change. So what happens is that when the blue form goes on to interact with the protein, in order to maintain that equilibrium, some of that red form begins to convert into the blue form. And that is what causes the color change in the Bradford assay. We move towards more of a blue hue. Now what we do is that we take our protein sample, we add in our Bradford reagent into our cuvette, and then we measure it in a spectrophotometer at about 460 nanometers and at around 600 nanometers. But what's important to understand is that the Bradford assay, it is composed of this Bradford reagent, which has this dye that exists in 3 different forms. And these 3 forms must be maintained in equilibrium. And when the blue form goes on to interact with the protein, that red form, some of it is converted into the blue form to maintain the equilibrium. And as a result, we move towards a blue color. So of course, we can make the understanding that the more protein we have, the more of the blue form of the dye will interact with the protein, resulting in higher absorbance and a more intense blue color. And that is essentially the Bradford assay. Once again, it is a colorimetric protein assay in which we are utilizing a dye molecule, dye-based molecule interaction with our protein to result in an absorbance value which we can quantify.